In 2008, a research team at the Shanghai Institute of Endocrinology and Metabolism did something that should have made international headlines. They took 116 patients with newly diagnosed type 2 diabetes. They split them into two groups. One group received metformin, the most prescribed diabetes drug on earth, the gold standard of blood sugar management, the medication that 150 million people take every single day. The other group received a compound extracted from the root bark of a yellow-flowered shrub that had been growing in Chinese medicine gardens for 3,000 years. The trial ran for 13 weeks. At the end of it, the researchers published their results in the peer-reviewed journal Metabolism. What they found did not compute with anything the pharmaceutical industry had built its diabetes franchise on. The plant compound reduced fasting blood glucose by 20%. Metformin reduced it by 23%. The plant compound reduced HbA1c, the 3-month blood sugar average that determines long-term diabetes prognosis, by 18%. Metformin reduced it by 17.5%. For triglycerides, for LDL cholesterol, for post-meal blood sugar spikes, the plant compound did not fall short of the drug. In several markers, it outperformed it. The compound was berberine. The plant was a common barberry shrub. It cost $9 for a 1-month supply. Today, not one of the 37.3 million Americans living with type 2 diabetes will hear that number from their endocrinologist. Their doctor will not write a berberine prescription. Their pharmacist will not stock the pharmaceutical-grade formulation. Their insurance will not cover it. Their medical textbook dedicated exactly 0 pages to it. But in China, it has been a registered pharmaceutical drug since 1956. In Germany, it sits in naturopathic formularies beside conventionally approved compounds. Across India, Ayurvedic physicians have documented its clinical use for longer than Western medicine has existed as a discipline. Not because the evidence is weak, because the evidence is unpatentable. Welcome to Seed Time Botanics. If this is your first time here, subscribe before we go any further and hit the bell. Because what we are about to show you about berberine, the history, the mechanism, the studies they never cited in your doctor's office, is the kind of information that this channel exists to put in your hands. Every video we make is built on the same principle. The seed always knows. The system just stopped telling you to plant it. The archive opens in 650 BC. A Chinese physician named Yin, credited among the earliest formulators of what would become traditional Chinese medicine, recorded the use of huang lian, the Chinese gold thread plant containing berberine, for conditions described in classical texts as Ri-Du Xia Li, heat toxicity with intestinal disorder. The modern translation of that clinical presentation is bacterial diarrhea with systemic inflammation. He prescribed the root extract, and he documented what happened when he did. He was not guessing. He was recording a result that practitioners before him had already been producing. In 200 BC, the Shinong Benchao Jing, the Divine Farmer's Classic of Materia Medica, the foundational pharmacological encyclopedia of Chinese medicine, listed huang lian as a first-class herb. Not supplementary. First class. Reserved for compounds that extended life, prevented illness, and demonstrated repeatable therapeutic effect across populations. The Ayurvedic tradition was building the same case from a different shrub. Berberis aristata, the Indian barberry, known in Sanskrit as Daraharidra, appeared in the Charaka Samhita around 600 BC as a remedy for skin conditions, eye infections, liver disorders, and what the text described as sugar in the urine. Sugar in the urine is the ancient clinical description of diabetes. They did not have blood glucose meters. They did not have HbA1c assays. But they tasted the urine of symptomatic patients, which sounds appalling and was, in the context of their era, a diagnostic protocol. And they observed that the barberry root extract changed what they found. The sugar diminished. The symptoms resolved. In the medieval Islamic tradition, Ibn Sina, Avicenna, included barberry preparations in the canon of medicine in 1025 AD as a treatment for liver disease, inflammation, and fevers. His pharmacological encyclopedia was the primary medical reference text in European universities for 500 years. The plant crossed into European botanical medicine through the trade routes. Nicholas Culpeper, in his 1653 Complete Herbal, described barberry bark as good to cool the heat of the liver and to open its obstructions. The language is pre-scientific. The clinical observation is not. 3,000 years. Across Chinese, Ayurvedic, Islamic, and European traditions simultaneously, independently, without communication between them. The same plant, the same conditions, the same recorded results. Then in 1917, a team of biochemists first isolated the pure compound from the plant material and gave it the name berberine. And the modern science of what 3,000 years of observation had been seeing in the dark, without microscopes, without molecular imaging, without randomized controlled trials, finally had the tools to understand what was actually happening. The reason berberine matched metformin in that 2008 Shanghai trial is not coincidence, and it is not placebo effect, and it is not scientific error. It is because both compounds activate the same cellular switch. The switch is called AMPK, adenosine monophosphate activated protein kinase. It is one of the most critical regulatory enzymes in human metabolism. Think of AMPK as the master energy sensor of every cell in your body. It also directly inhibits the enzyme alpha-glucosidase in the small intestine, the enzyme that breaks complex carbohydrates into glucose for absorption. By partially blocking this enzyme, berberine slows the rate of glucose entry into the bloodstream after meals. This is the same mechanism as the diabetes drug class called alpha-glucosidase inhibitors, which are sold under brand names like acarbose. So berberine does what metformin does, and what acarbose does, and it modifies the gut microbiome simultaneously, and it reduces LDL cholesterol through a mechanism similar to statin drugs. In a single compound, from a root bark that costs $9 a month. China did not wait for Western regulatory consensus. In 1956, the Chinese Pharmaceutical Authority approved berberine hydrochloride as a prescription drug for bacterial gastroenteritis under the name Huanglian Su. It appeared in the Chinese Pharmacopoeia, the national drug standard equivalent to the U.S. FDA's drug formulary, and has remained there in every subsequent edition. But the Chinese medical establishment did not stop at gastroenteritis. As the evidence base for berberine's metabolic effects accumulated through the 1990s and 2000s, Chinese endocrinologists began prescribing it off-label and then on-label for type 2 diabetes, dyslipidemia, and metabolic syndrome. Today, Chinese clinical practice guidelines formally recognize berberine as an adjunct therapeutic option for type 2 diabetes management alongside metformin and insulin. In India, the regulatory framework for Ayurvedic medicine formally includes berberine-containing preparations under the Drugs and Cosmetics Act. Berberis aristata extract is prescribed by qualified Ayurvedic practitioners for diabetes, liver disease, and inflammatory conditions with the same regulatory standing as pharmaceutical compounds within that system. In Europe, berberine preparations are available as registered botanical medicines in Germany, France, and Italy under the European Traditional Herbal Medicinal Products Directive. While this classification is not equivalent to full pharmaceutical prescription status, it represents regulatory recognition of the evidence base, something the American system has explicitly not provided. The global picture is consistent. Every major medical tradition that has had 3,000 years to observe this compound has found the same thing. The one that has had the compound for 30 years and chose to classify it as a supplement alongside protein powder and vitamin gummies is the outlier. Let us be precise about what the evidence says, because precision is the standard this channel holds itself to. The 2008 Zhang et al. Trial in Metabolism is the foundational human comparison study. 116 patients, 13 weeks, berberine versus metformin, equivalent outcomes across fasting glucose, HbA1c, post-meal glucose, triglycerides, and LDL cholesterol. This study has been cited in peer-reviewed literature over 800 times. A 2012 meta-analysis published in the Journal of Ethnopharmacology reviewed 14 randomized controlled trials involving 1,068 patients. It found that berberine combined with lifestyle intervention produced significantly greater reductions in fasting blood glucose and HbA1c than lifestyle intervention alone. It found that berberine combined with diabetes medication produced significantly greater reductions than medication alone. The authors described the evidence as supporting berberine's role as a clinically meaningful adjunct in diabetes management. A 2023 systematic review and meta-analysis in Frontiers in Pharmacology, published just 2 years ago, analyzed 46 randomized controlled trials involving 4,851 participants. It confirmed significant reductions in fasting blood glucose, post-meal glucose, HbA1c, body weight, waist circumference, total cholesterol, LDL cholesterol, and triglycerides. It found the safety profile comparable to metformin, with the primary side effect being mild gastrointestinal discomfort in a minority of users, the same side effect profile that metformin carries. A 2023 trial published in Phytomedicine specifically examined berberine in patients who were already insulin-resistant but not yet diabetic. The pre-diabetic population of 96 million Americans whose progression to type 2 diabetes could potentially be slowed or prevented. The berberine group showed significant improvements in insulin resistance markers compared to placebo after 12 weeks. Across those 3,000 studies, the signal is consistent. The safety profile is documented. The mechanism is understood. The clinical results are replicable. The word for a compound with this profile in the pharmaceutical world is approved. In the American supplement world, it is not evaluated by the FDA. This is the part of the video where the picture starts to become very clear and very uncomfortable. If you have not subscribed yet, do it now and share this with someone you know who is managing blood sugar. Because what comes next is the structural reason, the specific, documented, financial reason why your doctor has never mentioned berberine in a single consultation. And once you see the architecture of it, you cannot unsee it. In 1994, the United States Congress passed the Dietary Supplement Health and Education Act. The legislation was framed as a protection of consumer freedom, a guarantee that Americans could access vitamins, herbs, and natural compounds without excessive government restriction. It was presented as a victory for natural medicine. It was structurally a quarantine. Because of DSHEA, berberine cannot be regulated as a drug in the United States without a company investing approximately $800 million to $1.2 billion in the clinical trial program the FDA requires for new drug applications. That investment only makes financial sense if the company can patent the compound at the end of it. If they can own the exclusive right to sell the approved product for a defined period and recoup the development cost through protected market share. You cannot patent berberine. It is a natural compound found in multiple plant species across multiple continents. It has been documented in scientific literature for over a century. No pharmaceutical company can own it. So no pharmaceutical company will spend the $1 billion to prove it in the specific way the FDA accepts as proof. And because no company has spent the $1 billion, the FDA cannot approve it. And because the FDA has not approved it, American physicians cannot prescribe it. And because American physicians cannot prescribe it, medical schools do not train physicians to recommend it. And because medical schools do not train physicians to recommend it, the 37.3 million Americans with type 2 diabetes will never hear about it in the office where they receive their diabetes management. The circle is perfect, and it is not an accident. But there is a second layer to the structural failure, and it directly undermines the clinical evidence that does exist from American research. Without FDA oversight, American manufacturers of berberine supplements have no obligation to standardize the compound's concentration, no obligation to verify that the capsule contains what the label states, no obligation to use pharmaceutical-grade extraction methods. A 2021 independent laboratory analysis of commercially available berberine supplements in the American market found potency variation of up to 60% between products, meaning the capsule labeled as 500 milligrams might contain anywhere from 200 milligrams to 750 milligrams of actual berberine hydrochloride. The American clinical trials that have produced inconsistent results for berberine were largely conducted using supplement-grade material of unverified potency. The Chinese trials that produced the metformin equivalent results used pharmaceutical-grade standardized berberine hydrochloride at confirmed concentrations. The system arranged the conditions of its own negative evidence, and then cited that evidence as the reason not to act. The global diabetes drug market was valued at $67.3 billion in 2023. It is projected to reach $132.7 billion by 2031. The GLP-1 receptor agonist class, Ozempic, Wegovy, Mounjaro, Trulicity, generated over $50 billion in combined revenue in 2023 alone. Novo Nordisk's market capitalization briefly exceeded the entire GDP of Denmark on the strength of semaglutide sales. A one-month supply of Ozempic without insurance costs between $900 and $1,000 in the United States. Global shortages have been documented across multiple countries as manufacturing cannot meet prescription demand. These are remarkable drugs. Their mechanisms are documented. Their clinical results in weight loss and blood sugar management are real. This is not an argument against their efficacy. It is a question about their necessity as the singular first-line response to a metabolic crisis that a $9 plant compound with a 3,000-year documented history and 3,000 peer-reviewed studies has been addressing, quietly, effectively, and without the $900 monthly invoice, for longer than Western medicine has existed. The 96 million pre-diabetic Americans who could potentially arrest their progression before the first metformin prescription are not being told about berberine. They are on a trajectory toward a $132.7 billion market that requires their continued disease progression to sustain its valuation. This is not a conspiracy. It is a structure. Structures do not require conspiracy. They require only that every participant follow the financial incentives built into them, and the outcome produces itself. In 2012, a clinical team at Gwangyuan Hospital in Beijing completed a 12-week trial specifically designed to assess berberine in patients with metabolic syndrome. The combination of high blood sugar, high blood pressure, abdominal obesity, and abnormal cholesterol that precedes full type 2 diabetes in millions of patients. They enrolled 37 patients. At 12 weeks, the berberine group showed an average fasting glucose reduction of 22.5%, a waist circumference reduction of 3.4 centimeters, significant improvements in all lipid markers, and reduction in inflammatory markers, including C-reactive protein. Not one of these patients was in a country where their primary care physician would suggest berberine at the point of metabolic syndrome diagnosis. Not one had a health insurer that would cover it. Every one of them was on a trajectory toward a metformin prescription and then potentially toward an incretin drug, a statin, and an antihypertensive, a 4-drug combination that costs between $300 and $600 per month at American pharmaceutical pricing. A 2020 review published in Oxidative Medicine and Cellular Longevity examined berberine's effects across diabetic patients already experiencing kidney stress, diabetic nephropathy, the leading cause of kidney failure in the developed world. It found that berberine reduced markers of kidney damage, reduced albuminuria, and exerted protective effects on kidney tissue through anti-inflammatory and antioxidative pathways. No approved diabetes drug currently on the American formulary addresses kidney protection as a primary mechanism in the same cost range. Share this video before we get to the practical section. Not for the algorithm, because someone in your life is managing blood sugar right now and has never heard a single word about what you have just learned. Put it in front of them. That is what this channel is for. Here is what the clinical evidence allows Seedtime Botanics to tell you with documented confidence. The formulation used in the successful comparison trials is berberine hydrochloride, not berberine sulfate, not berberine complex, berberine hydrochloride, standardized to a minimum of 97% purity. The dosage consistently used across successful trials is 500 milligrams taken 3 times daily with meals. The 3 times daily protocol is not optional. Berberine has a short half-life in the body, approximately 4 hours, which means a single daily dose produces a peak and a drop rather than the sustained metabolic effect the trials were measuring. Research confirms that berberine produces its most significant metabolic effects when used early in the disease trajectory, in prediabetic and early-stage diabetic patients whose pancreatic function is still intact. The AMPK activation mechanism is most effective when the cellular machinery it is supporting has not yet been significantly degraded. This is the same principle the Seedtime Botanics channel is built on. The plant works best when you plant the seed before the drought arrives. 3,000 years of documented clinical use. 3,000 peer-reviewed studies. A head-to-head trial against the most prescribed diabetes drug on Earth that produced equivalent outcomes. Prescription drug status in China, regulatory recognition across India, Europe, and Asia. A safety profile across those 3,000 studies that is comparable to the drug it matched in the trial. And 37.3 million Americans with type 2 diabetes whose physicians have not mentioned it once. The global diabetes drug market is approaching $133 billion. The compound that matched its gold standard in controlled conditions costs $9 a month and grows as a shrub in USDA zones 3 through 9. The knowledge was never hidden. The mechanism was isolated in 1917. The head-to-head trial was published in a peer-reviewed journal in 2008. The 2023 meta-analysis covering nearly 5,000 patients is available to any physician with a research database login. It was not lost. It was not disproven. It was classified as a supplement in 1994 by a law that removed every financial incentive for the clinical investment that American regulatory approval requires, and then the absence of that approval was cited as the reason not to use it. The barberry shrub still grows in hedgerows across the American Northeast. Oregon grape still covers the forest floor in the Pacific Northwest. The yellow root is still there. The compound is still in it. The mechanism still activates AMPK in exactly the same way it did in the Shanghai trial, and in the Beijing metabolic syndrome study, and in the 3,000 studies that preceded this video. The seed always knew. The system just stopped telling you to plant it. If this vault opened something for you today, subscribe to Seedtime Botanics, hit the bell, and share this with one person managing blood sugar who has never heard the word berberine in a medical consultation. Every plant shares the seed somewhere the system did not intend it to grow. The next vault opens soon.